In many ways, progress in biology over the past century is reflected in our changing concept of a gene. As the result of Mendel's work, biologists learned that genes are discrete elements that govern the appearance of specific traits. Given the opportunity, Mendel might have argued for the concept that one gene determines one trait. Boveri, Weismann, Sutton, and their contemporaries discovered that genes have a physical embodiment as part of the chromosome. Morrigan and colleagues demonstrated that genes have specific addresses. They reside at particular locations in particular chromosomes, and these addresses remain constant from one individual of a species to the next. Griffith, Avery, and Hershey and Chase demonstrated that genes were composed of DNA, and Watson and Crick solved the puzzle of DNA structure. It explained how this remarkable macromolecule could encode heritable information. Although the formulations of these concepts were milestones along the path of genetic understanding, none of them addressed the mechanism by which the information stored in a gene is put to work in governing cellular activities. This is the major topic to be discussed in the present chapter. We will begin with additional insights into the nature of a gene, which brings us closer to its role in the expression of inherited traits. So, first topic, the relationship between genes, proteins, and RNAs. The first meaningful insight into gene function was gained by Archibald Gerald, a Scottish physician who reported in 1908 that the symptoms exhibited by persons with certain rare inherited diseases were caused by the absence of specific enzymes. One of the diseases investigated by Gerald was alkaptonuria, a condition readily diagnosed because of the urine becomes dark on exposure to air. Gerald found that persons with alkaptonuria lacked an enzyme in their blood that oxidized homogenistic acid, a compound formed during the breakdown of amino acids phenylalanine and tyrosine. As homogenistic acid accumulates, it is excreted in the urine and darkens in color when oxidized by air. Gerald had discovered the relationship between the genetic defect, a specific enzyme, and a specific metabolic condition. He called this disease inborn errors of metabolism, as seems to have happened with other early observations of basic importance in genetics. Gerard's findings went unappreciated for decades. The idea that genes direct the production of enzymes was reassured in 1940 by George Bade and Edward Tatum of California Institute of Technology. They studied Neurospora, a tropical bread mold that grows in a very simple medium containing a single organic carbon source, inorganic salts, and biotin, a B vitamin, because it needs so little to live on. Neurospora was presumed to synthesize all of its required metabolites. Badel and Tatum reasoned that an organism with such broad synthetic capacity should be very sensitive to enzymatic deficiencies, which should be easily detected using proper experimental protocol. A simplified outline of their protocol is very important, and Bedline and Tatum plan was to irradiate mold spores and screen them for mutations that caused cells to lack a particular enzyme. To screen for such mutations, irradiated spores were tested for their ability to grow in a minimal medium that lacked the essential compounds known to be synthesized by this organism. If a spore is unable to grow in minimal medium, but a genetically identical spore can grow in a medium supplemented with a particular coenzyme, pantothenic acid of coenzyme A, then the researchers could conclude that the cells have an enzymatic deficiency that prevents them from synthesizing this essential compound. Badel and Tatum began by irradiating over 1,000 cells. 2 of the spores proved unable to grow on the minimal medium. One needed pyridoxine, vitamin B6, and other needed thiamine, vitamin B1. Eventually, the progeny of about 100,000 irradiated spores were tested. The dozens of mutants were isolated. Each mutant had a gene defect that resulted in an enzyme deficiency that prevented the cells from catalyzing a particular metabolic function. The results were clear-cut. The gene carries the information for the construction of a particular enzyme. This conclusion became known as one gene, one enzyme hypothesis. Once it was learned that enzymes are further composed of more than one polypeptide, each of which is encoded by its own gene, and the concept becomes modified to one gene, one polypeptide hypothesis. Although this relationship remains a close approximation of the basic functions, that a single gene often generates a variety of polypeptides, primarily as a result of alternative splicing. It would also become apparent that describing a gene strictly as an information store for polypeptides is far too that, rather than containing information for polypeptide synthesis, function as RNAs in their own right. With this in mind, it might be best to define a gene as a segment of DNA that contains the information for either a single polypeptide chain or for one or more functional RNA. Then, an overview of the flow of information through the cell. The information present in a segment of DNA is made available to the cell through formation of a molecule of RNA. The synthesis of an RNA from DNA template is called transcription. The term transcription denotes a process in which the information encoded in the 4-deoxyribonucleotide letters of DNA is rewritten or transcribed into a similar language composed of 4 ribonucleotide letters of RNA. We will examine the mechanism of transcription shortly. But first, we will continue with the role of gene in the formation of polypeptides. Studies carried out in 1950s uncovered the relationship between genetic information and amino acid sequence, but this knowledge by itself provided no clue as to the mechanism by which the specific polypeptide gene is generated. As we know now, there is an intermediate between a gene and its polypeptide, and the intermediate is messenger RNA, mRNA. The momentous discovery of mRNA was made in 1961 by Francis Jacob Monod of the Pasteur Institute in Paris, Sidney Brenner of the University of Cambridge, and Matthew Michelson of California Institute of Technology. A messenger RNA is assembled as a complementary copy of one of the 2 DNA strands that make up a gene. Because its nucleotide sequence is complementary to that of the gene from which it is transcribed, the mRNA retains the same information for polypeptide assembly as the gene itself. For this reason, an mRNA can also be transcribed as a sense strand or a coding RNA. Eukaryotic mRNA are not synthesized in their final or mature form, but instead must be carved out or processed from much larger pre-mRNA. An overview of the role of mRNA in the flow of information is very important. The use of messenger RNA allows the cell to separate information storage from information utilization. While the genes remain stored away from the nucleus as part of a huge stationary DNA molecule, its information can be imparted to a much smaller mobile nucleic acid that passes into the cytoplasm. Once in the cytoplasm, the mRNA can serve as a template to direct the incorporation of amino acids in a particular order encoded by the nucleotide sequence of the DNA and mRNA. The use of messenger RNA also allows the cells to greatly amplify its synthetic output. One DNA molecule can serve as a template in the formation of many mRNA molecules, each of which can be used in the formation of a large number of polypeptide genes. Then proteins are synthesized in the cytoplasm by a complex process called translation. Translation requires the participation of dozens of different components, including ribosomes. Ribosomes are complex cytoplasmic machines that can be programmed like a computer to translate the information encoded by any mRNA. Ribosomes contain both protein and RNA. The RNA of a ribosome are called ribosomal RNAs or rRNAs, and like mRNAs, each is transcribed from one of the DNA strands of a gene. Rather than functioning in an informational capacity, rRNA provides structural support and catalyze the chemical reaction in which amino acids are covalently linked to one another. Transfer RNA or tRNA constitute a third major class of RNA that is required during protein synthesis. Transfer RNAs are required to translate the information in the mRNA nucleotide code into the amino acid alphabet of a polypeptide. Both rRNA and tRNA owe their activity to their complex secondary and tertiary structures. Unlike DNA, which has a similar double-stranded helical structure regardless of a source, many RNAs fold into a complex 3-dimensional shape which is markedly different from one type of RNA to another. Thus, like proteins, RNAs carry out a diverse array of functions because of their different shapes. As with proteins, the folding of RNA molecules follows certain rules. Whereas protein folding is driven by withdrawal of hydrophobic residues into the interior, RNA folding is driven by the formation of regions having complementary base pairing. Base pairing regions typically form double-stranded and double helical stems which are connected to single-stranded loops. Unlike DNA, which consists of exclusively of stranded Watson and Crick base pairs, RNAs often contain non-stranded base pairs and modified nitrogenous bases. This unorthodox regions of the molecule serve as recognition sites for proteins and other RNAs, promote RNA folding, and help stabilize the structure of the molecule. The importance of complementary base pairing extends far beyond tRNA and rRNA structure. As we will see through this chapter, base pairing between RNA molecule plays a central role in most of the activities in which RNAs are engaged. The roles of mRNAs, rRNA, and tRNA are explored in detail in the following sections of this chapter. Eukaryotic cells make a host of other RNAs which also play vital role in cellular metabolism. This includes small nuclear RNAs, snRNA, small nuclear RNA, snORNA, small interfering RNA, siRNA, piRNA, microRNA, and miRNA, and a variety of other non-coding RNAs, that is RNA that do not contain information for amino acid sequence. Most of these RNAs serve as regulatory function, controlling various aspects of gene expression. Several of these RNAs have burst onto the scene in the past few years, reminding us once again that there are many hidden cellular activities waiting to be discovered. Next topic, synthesis and processing of eukaryotic ribosomal and transfer RNA. A eukaryotic cell may contain millions of ribosomes, each consisting of several molecules of rRNA together with dozens of ribosomal proteins. The composition of a mammalian ribosome is shown: 49 ribosomal proteins, 33 ribosomal proteins, and 5s rRNA, 5.8s rRNA, 18s rRNA, 28s rRNA, 60s subunit, 40s subunit, etc. are present. Ribosomes are so numerous that more than 80% of the RNA in most cells consist of ribosomal RNA. To furnish the cell with such a large number of transcripts, the DNA sequence encoding rRNA are normally repeated hundreds of times. This DNA, called rDNA, is typically clustered in one or a few regions of the genome. The human genome has 5 rRNA clusters, each on a different chromosome. In a non-dividing interphase cell, the clusters of rDNA are gathered together as part of one or more regularly spaced nuclear structures called nucleoli, singular nucleolus, that function in producing ribosomes. The bulk of nucleolus is composed of nascent ribosomal subunits that give the nucleolus a granular appearance. Embedded within this granular mass are one or more rounded cores consisting primarily of fibrillar material. Synthesizing the rRNA precursor. Oocytes are typically very large cells. Those of amphibians are generally enormous, up to 2.5 mm in diameter. During the growth of amphibian oocytes, the amount of rDNA in the cell is greatly increased, as are the number of nucleoli. The selective amplification of rDNA is necessary to provide the large amount of ribosomes that are required by the fertilized egg to begin embryonic development. Because these oocytes contain hundreds of nucleoli, each actively manufacturing rRNA, they are ideal subjects for the study of rRNA synthesis and processing. Our understanding of rRNA synthesis and DNA transcription in general was greatly advanced in the late 1960s with the development of techniques by Oscar Miller Jr. of the University of Virginia to visualize genes in action with the electron microscope. To carry out these studies, the fibrillar cores of oocyte nucleoli were gently dispersed to reveal the presence of a large circular fiber. When one of these fibers was examined in the electron microscope, it was seen to resemble a chain of Christmas trees. The electron micrographs and the drawings are very important, and they reveal this nucleolar activity and rRNA structure synthesis. Then processing of rRNA precursor. Eukaryotic ribosomes have 4 distinct ribosomal RNAs, 3 in the large subunit and 1 in the small subunit. In humans, the large subunit contains a 28S, 5.8S and 5S RNA molecules, and the small subunit contains an 18S RNA molecule. 3 of these rRNAs, the 28S, 18S and 5.S, are carved by various nucleases from a single primary transcript called the pre-rRNA. The 5S RNA is synthesized from a separate RNA precursor outside the nucleus. 2 of the peculiarities of pre-rRNA as compared with other RNA transcripts are the large number of methylated nucleotides and pseudouridine residues. By the time a human pre-rRNA precursor is first cleaved, over 100 methyl groups have been added to ribose groups in the molecule, and approximately 95% of the uridine residue have been enzymatically converted to pseudouridine. All of these modifications occur after the nucleotides are incorporated into the nascent RNA, that is, post-transcriptionally. The altered nucleotides are located at specific positions and are clustered within portions of the molecule that have been conserved among organisms. All of the nucleotides in the pre-rRNA that are altered remain as part of the final products, while unaltered sections are discarded during processing. The functions of the methyl groups and pseudouridines are unclear. These modified nucleotides may protect parts of pre-mRNA from enzymatic cleavage, promote folding of rRNA into their final 3-dimensional structures, or promote interaction of rRNA with other molecules. Because rRNA are now heavily methylated, their synthesis can be followed by incubating cells with radioactively labeled methionine, and compound that serves as most cells in a methyl group donor. The methyl group is transferred enzymatically from methionine to nucleotides in the pre-mRNA when 14C methionine is provided to cultured mammalian cells for a short period of time. A considerable fraction of the incorporated radioactivity is present in the 45S RNA molecule, equal to a length of about 13,000 nucleotides. A 45S RNA is cleaved into smaller molecules that are then trimmed down to the 28S, 18S and 5.8S rRNA molecules. The combined length of these 3 mature rRNAs is approximately 700 nucleotides, or a little more than half of the primary transcript. Some of the steps in the processing pathway from the 45S pre-rRNA to the mature rRNA can be followed by incubating mammalian cells very briefly with labeled methionine, and then raising the cells in non-labeled medium for varying lengths of time. The step of pulse cheese experiment was very important, and as noted above, the first species to become labeled in this type of experiment is the 45S primary transcript, which is seen as a peak of radioactivity and nuclear RNA function after 10 minutes. After about an hour, the 45S RNA has disappeared from the nucleolus and is largely replaced by a 32S RNA, which is one of the 2 major products produced from the 45S primary transcript. The 32S RNA is seen as a distant peak in the nuclear fraction from 40 minutes to 150 minutes. The 35S RNA is a precursor to the mature 28S and 5.8S rRNA. The other major product of 45S pre-rRNA leaves the nucleolus quite rapidly and apparently in the cytoplasm as the mature 18S rRNA. After a period of 2 or more hours, nearly all of the radioactivity has left the nucleolus and most has accumulated in the 28S and 18S rRNAs of the crypt cytoplasm. The radioactivity in the 5S rRNA peak include the 5.8S RNA as well as methyl groups that have transferred to small tRNA molecule. Then the role of SNO RNAs. The processing of the pre-RNA is accomplished with the help of a large number of small nuclear RNA or snRNA that are packaged with particular proteins to form particles called snORNPs, small nuclear ribonucleoproteins. Electron micrographs indicate that snORNPs begin to associate with the rRNA precursor before it is fully transcribed. The first RNP particle to attach to pre-rRNA transcript contains the U3SNORNA and more than 2 dozen different proteins. This huge component of rRNA processing machinery can be seen as a ball bound to the outer end of each nascent RNA fibril in this figure. So, when it is catalyzed, the removal of 5' end of the transcript, some of the other enzymatic cleavages are thought to be catalyzed by the exosome, which is an RNA binding machine that consists of nearly a dozen different exonucleases. U3 and 7 other snORNAs were identified years ago because they are present in large quantities, about 10 raised to 6 copies per cell. Subsequently, a different class of snORNAs present at lower concentrations, about 10 raised to 4 copies per cell, was discovered. This low abundance snORNAs were then divided into two groups based on their function and similarities in nucleotide sequence. Members of one group, called the box C by D snORNAs, determine which nucleotides in the pre-RNAs will have their ribose moieties methylated, whereas members of other group, called the box H to ACA snORNAs, determine which uridine are converted to pseudouridine. The structure of the nucleotides modified by whose reactions are very important. Then, both groups of snORNAs contain relatively long stretches that are complementary to sections of the rRNA transcript. These snORNAs provide an excellent example of the principle that single-stranded nucleic acids having complementary nucleotide sequences are capable of forming double-stranded hybrids. In this case, each snORNAs bind to a specific portion of the pre-RNA to form an pre-RNA-RNA duplex. The bound snORNA then guides an enzyme, either a methylase or a pseudouridylase, within the snRNAs or modify a particular nucleotide in the pre-rRNAs. Taken together, there are roughly 200 different snORNAs, one for each site in the pre-rRNA, that is ribose methylated or pseudouridinated. If the DNA encoding one of these snORNAs is deleted, one of the nucleotides of the pre-rRNAs fails to be enzymatically modified. The mechanism of action of the two types of snORNA is very important. The nucleolus is the site not only for rRNA processing but also assembling of the two ribosomal subunits. More than 200 different proteins have been found to be associated with the rRNA at different stages of ribosome assembly. These proteins can be divided into two groups: ribosomal proteins that remain in the subunit and accessory proteins that have a transient interaction with the rRNA intermediates and are required only for processing. Among this later group are more than a dozen RNA helicases, which are enzymes that unwind regions of double-stranded RNA. These enzymes are presumably involved in many structural rearrangements that occur during ribosome formation, including the dissociation of snORNAs from pre-ribosomal particles, synthesis and processing of 5S rRNA. The 5S rRNA, about 120 nucleotides long, is present as part of a large ribosomal subunit of both prokaryotes and eukaryotes. In eukaryotes, the 5S rRNA molecule are encoded by a large number of identical genes that are separate from the other rRNA genes and are located outside the nucleolus. Following this synthesis, the 5S rRNA is transported to the nucleolus to join the other components involved in the assembly of ribosomal subunits. The 5S rRNA genes are transcribed by RNA polymerase III. RNA polymerase III is unusual among the three polymerases in that it can bind to a promoter site located within the transcribed portion of the target gene. The internal position of the promoter was first clearly demonstrated by introducing modified 5S rRNA genes into host cells and determining the ability of that DNA to serve as a template for the host polymerase III. It is found that the entire 5' flanking region could be removed and the polymerase would still transcribe the DNA starting at the normal initiation site. If, however, the deletion included the central part of the gene, The polymerase would not transcribe the DNA or even bind to it. If the internal promoter from a 5S rRNA gene is inserted into another region of the genome, the new site becomes a template for transcription by RNA polymerase III. Then transfer RNAs. Plant and animal cells are estimated to have approximately 50 different species of transfer RNA, each encoded by a repeated DNA sequence. The degree of repetition varies with the organism. Yeast cells have a total of about 275 tRNA genes, fruit flies about 850, and humans about 1300. Transfer RNAs are synthesized from genes that are found in small clusters scattered around the genome. A single cluster typically contains multiple copies of different tRNA genes, and conversely, the DNA sequence encoding a given tRNA is typically found in more than one cluster. The DNA within a cluster, or a tDNA, consists largely of non-transcribed base sequences with the tRNA coding sequence situated at irregular intervals in a tandemly repeating arrangement. Like the 5S rRNA, tRNAs are transcribed by RNA polymerase III, and promoter sequence lies within the coding section of the gene rather than being located at its 5' flank. The primary transcript of a transfer RNA molecule is larger than the final product. Pieces of both the 5' and 3' sides of the precursor tRNA and a small interior piece in some cases must be trimmed away. In addition, numerous bases must be modified. One of the enzymes involved in pre-tRNA processing is an endonuclease called ribonuclease P, which is present in both bacterial and eukaryotic cells and consists of both RNA and protein subunits. It is the RNA subunit of ribonuclease P that catalyzes cleavage of the pre-tRNA substrate as substrate. Continuation of this topic will be dealt with the help of next video.